So, a very good morning to all of you. Today is the last date for this workshop. So, actually, we are supposed to come close our workshop in yesterday itself. But even though, as per the Gyan norms, we are supposed to conduct exam. Okay. So, before completion of our lecture schedule, we cannot conduct exams because exam will be based on all these things. Okay, all the lectures must be covered, and based on all the lectures we need to conduct exam. Okay, so I don't know whether you know or not. It's having some credit also. It's a 1 credit course. It looks like. Okay, so BTech students or MTech students, they can use if your college having some norms like this many credits you have to earn, means you have to show this one as I have earned 1 credit in this. Okay, so this is a policy of Gyan. Only last time it is not like this. Okay, this time they have followed this 1 credit course. Okay, 10 to 14 lectures. Okay, if you attend, it will be the 1 credit course. So 24 to 28 lectures if you attend, it will be 2 credit course. Okay, then based on the that exam paper, we need to assign your grade also. Grade means like either S grade or A grade or B grade and all, we need to do that. Okay. So yesterday itself I have informed about that. Today after these sessions we have examination. Okay, you have to participate. Even either the offline participants or online participants, so they have to attend the exams. Okay. So okay, so before starts my presentation, so we have seen yesterday two topics. One is the bioelectrochemical system for the electricity production, and the second one is the bioelectrochemical system for the it's a hydrogen productions. Okay, so these I have given you some concept of all these things, these 2 topics. Okay, so some experimental results also I have shown in the yesterday's presentation. So that work was done by my PhD scholar. Okay, so Dr. Madhesan has shown one results. That is that work was done by my PhD scholar Dr. Tamil Mani. So then another work. Okay, so which was done by my scholar, PhD scholar, Mrs. Jayachitra, and few more works done by my MTech students also. So I thank my scholars. Okay, so now some part of the work I am going to show in this. Okay, that is also done by one of my PhD scholar, Mrs. Sandhya. Okay, Mrs. Sandhya and other students also. Okay, so I will show that results. Okay, fine. This is one of the topic. Okay, in addition to the whatever we covered in yesterday's. Bioelectrochemical system for water desalination and water remediation. Okay, up to yesterday we have up to yesterday we have seen this like bioelectrochemical system for only the wastewater treatment, and also it is a electricity and biohydrogen productions. Okay, so now we want to go for the desalination technology. Okay, desalination of the saline water. Okay, as you know this, there will be a 97% of the water will be the saline water, that is sea water. Okay, only 3% of the water only it is a drinkable water, so we can drink it. Okay, the rest of the 97% if you see it, almost so all the water contains it's a salt. Okay, salt either in the form of minerals. Okay, like if you see it, our groundwater level also it will be around 400 to the 500 ppm or 400 to 1000 ppm. Okay, depending upon the location. So if you drink that water, okay, so it will create lot of problems to the human body. Okay, maybe it may form the kidney stones, other effect may create in that. Okay, so many technologies available for treating this such kind of the water. Okay, or such kind of the water. Okay, like currently people are using this technology. One is thermal treatment, another one is the mechanical and the chemical treatment. Okay, these 3 methods generally used for the treatment of saline water. So if you see it like in our in a household, okay, we are using the RO systems. Okay, RO system is one kind of the process, one kind of the is a mechanical process. Okay, so via this we are treating that membrane. Okay, treating the saline water. Like here if you see it, but these are the technologies like thermal, mechanical, and the other kind of the treatment. Okay, it needs some kind of the energy to the systems. Okay, energy as well as, and some cases it will be harmful to the environment also. Okay, if you take that our systems, we are using the membrane. Okay, membrane is the one of the material for treating that saline water. Okay, so like if you use the membrane, okay, again it creates the environmental pollutions. Okay, so and also lot of problems with the membrane. So and efficiency also will be low as compared to the like another kind of the method. Okay, each method will have the as I told you yesterday itself, each method will have the some advantages and the disadvantages. So here the one of the technology is bioelectrochemical system. So this bioelectrochemical system is also used for treating the saline water, and also it is used for treating the wastewater along with energy generation. So here this bioelectrochemical system, we can say it is a microbial desalination cell. So microbial desalination cell is the combination of the two systems. Okay, one is that microbial fuel cell and another one is the electrodialysis. Okay, it's a combination of these two systems. So it is almost similar to the microbial fuel cell, but only the difference is that so it has one more chamber in between the anode and the cathode. So as per the microbial fuel cell, we have the two chamber, one is anode, another one is the cathode. So this anode and the cathode is separated by the proton exchange membrane. But as in the case of microbial desalination cell, which has three chamber. Okay, chamber 1 will be the anode and chamber 2 will be the cathode. So chamber 3, which will be in the middle, will be it is a desalination chamber. That is that chamber, okay, desalination chamber and anode chamber, which is separated by the anion exchange membrane. Similarly, it is a desalination chamber and the cathode chamber is separated by a cation exchange membrane. Okay, each will have the different function. Okay, so here if you see it, as you know, this is anode side, we are taking the wastewater. Okay, wastewater as a substrate, so microorganism as a catalyst. That microorganism will degrade the organic matter and it releases the carbon dioxide and the electrons and the protons. Okay, so similarly as in the case of cathode side, we have the oxygen, so we have the hydrogen will be transferred through this membrane and electron will be transferred through the external circuit to the cathode side. Okay, cathode side. So then what is the role of desalination chamber? So here we know this, it is a desalination chamber which contains salt water. Okay, salt means it is a NaCl solution. Okay, NaCl means it's a sodium chloride. Okay, that sodium chloride, what will happen? So there will be a concentration difference between these two chamber. Okay, these two chamber, because of the driving force, that is here we have the anion exchange membrane, that is negative ion transfer. Okay, anion. So whatever the anion is there, that it will transfer. Here it will be, if it is any positive ion, so cation or positive ion if it is there, then it will be transferred to this. Okay, so here what will happen, because of the driving force between the each chamber, okay, between this and similarly this and this, so here the chlorine ion will move to the anode side. Okay, similarly, so it's a sodium ion will move to the cathode side. So what will happen in this? So this chlorine ion will so this chlorine ion will move to the cathode side, and because of the driving force, it combined with the H plus ion, it form the HCl. Similarly, as in the case of here, it will be Na plus ion will move to the cathode side. Okay, it may form the NaOH solutions. Okay, sodium hydroxide solutions. So because of the driving force between the each chamber, so it is producing that so each chamber, so it produces because of the electron transfer, it produces the electricity, and because of the biodegradation, it removes the contaminant from the wastewater. At the same time, in the saline chamber, saline chamber, salt is also removed. Okay. So, these three process is taking place in the microbial desalination cell. So, and next one will be the, so that what are the components used in the microbial field cell or microbial electrolysis cell, it is that component will be the remain same, that means its electrode and all maybe remain same, but it depending upon the properties. Okay, like for the case of microbial fuel cell, we can use the same kind of the electrode in the both side, anode and the cathode side. But as in the case of microbial electrolysis cell, we cannot use the same electrode. So, because of in the anode side will be the oxidation reaction is taking place, cathode side will be the reduction reaction is taking place. Okay. So, if the reduction properties, if it is higher in the, if whatever the electrode you have selected, that electrode should have the reduction property. Okay. If it is reduction property, that electrode we can use it. Okay. Similarly here. Okay. So, that material electrode, whatever electrode you can choose it, that electrode should have the some important properties, like in the anode side if you use it, that should be the oxidation property. If it is in the cathode side, it should be the reduction characteristics. Okay. It can reduce the, it should reduce the ions. Okay. So, that property should be there. So, similarly here you can see lot of factors, okay, which will affect the performance of the microbial desalination cell. So, in the microbial desalination cell, there will be a wastewater concentration. Okay, wastewater concentration is one of the factor, and similarly the pH conditions of the analyte and the catholyte is another factor, and third one will be the desalination chamber. Okay, so saline concentration of the in the desalination chamber. Okay, that concentration is most important. Okay, so even if it's many factor, that salt water concentration plays a major role in the performance of the microbial desalination cell. So, why it is placing the major role in the microbial desalination cell? As I told you earlier, there will be a concentration driving force between the each chamber. Okay, anode to the desalination chamber, similarly desalination chamber to the cathode chamber. So, accordingly based on that only your ion transfer from one side to the another side. So, it is a concentration of the saline water will be the most significant as compared to the any other factors in this. So, this is one. So, here you can see this what will be the how the way the performance is improved and performance is decreased. Okay, here you can see. So, if it is I think is it visible or not, I do not know, it is a high salt concentration. If you increase the concentration, my performance is increasing. Okay, same time if I reduce the concentration of the saline water, so performance is decreasing. Similarly pH condition, similarly it is a what is a similarly like concentration and the internal resistance of the systems. So, most important factor is the saline water concentration. So, the next one will be the, so here like MDC performance. Okay. So, if I define the MDC performance will define in terms of what will be the wastewater treatment efficiency, and also it is a desalination efficiency, and third one will be the it is a energy production. Okay. Energy production either in the form of hydrogen or in the form of electricity. So, like MDC can be classified into different types. Okay, there is many types of MDC has been made. So, the first one will be the microbial electrodialysis cell. So, in the microbial electrodialysis cell, which is similar to the microbial desalination cell, the only the difference is, so it is also able to produce the hydrogen. Okay, instead of electricity, it can also able to produce the hydrogen. Okay, to produce the hydrogen, we need to supply the small potential to the system. So, here you can see it is this, it will be the, as in the case of microbial desalination cell will be the only 3 chamber. Okay, anode, desalination chamber and the cathode chamber. So, and the cathode chamber, but here this microbial electrodialysis cell, so it connect, so in the microbial desalination cell is connected to the external circuit. Okay, so for the hydrogen production, we need to connect this external circuit to the additional power supply. Okay, power source we need to connect it. Okay, to convert the hydrogen ion into the hydrogen molecules. Okay, it needs some kind of the energy to convert the hydrogen ion into the hydrogen molecule. This is a one, and another one is microbial electrodialysis and desalination cell. So, here it is nothing but, so in the previous one it is used for only the hydrogen production as well as the saline water treatment. Okay. So, here this one will be used for instead of hydrogen. Okay, instead of producing the hydrogen, you can produce some kind of the acid or base. Okay, like you can produce HCl if I want to produce HCl, or if I want to produce any other solutions, okay, from the MDC. Okay, so with the addition of power supply. Okay, this is another type, and another one will be the like similar to this microbial electrolysis cell, whatever I talked earlier, the same thing. Microbial electrolysis desalination cell. Okay, it is used for producing the hydrogen as well as the removal of salt water contained from the saline water. Okay, salt from the saline water. So here different studies have been carried out in this. So here you can see so how far this it is improving the performance with this. Okay, so like based on the saline water concentration or based on the wastewater concentration. Okay, so how far it is improving the performance. Okay, with respect to the conditions. Okay, so this is one another one. So as I told you in the yesterday's sessions. Okay, so this technology like microbial bioelectrochemical system that can be connected with the any kind of the existing systems, like it can be connected with the anaerobic digester or it can be connected with the dark fermentation. Okay, if we want to produce the hydrogen, we can connect the dark fermentation with microbial electrolysis cell. Okay, microbial electrolysis cell or microbial desalination cell. Similarly, if I want to produce the methane, at the same time we want to remove the saline water, remove the salt content from the saline water, so we can connect it with this MDC with the another systems. Okay, like it can be integrated with that. So here you can see, and another one is so as I told you, single cell cannot be produced cannot be produced more energy to the more energy for the real time applications. Okay, it can produce only very less voltage, so less voltage will not be enough for the real time applications. So we can go for the, instead of going for the single cell, we can go for the multiple cells, may be connected in the series or parallel. Okay, this is one type of stacked microbial desalination cell. So n number of cells may be connected together to form that. Okay, either the series or parallel depending upon the real time application. If I want to go for the higher energy, okay, higher energy, higher current needed for the systems, I have to connect the n number of cells in the parallel mode. So if I want to get the more voltage, okay, so more voltage means so we want to connect the all the cells will be in the parallel series manner. Okay, to get the voltage or energy. This is another type like upflow microbial desalination cell for the continuous mode of operations. Okay, for the continuous mode of operations. So here this is another type bipolar microbial desalination cell. So it is nothing but here it will be as I told you, like in the microbial desalination cell, anode is having the one electrode, anode side one electrode will be there, cathode side electrode will be there. So one electrode will be one electrode itself, it will act as a anode and the cathode side. Okay, so one side of the electrode will be the act as a anode, another side of the electrode will act as a cathode in this. Okay, this is another type. So now this is one of the different kind of the patent. Okay, so microbial desalination patent reported in the literatures. Okay, so several studies have been carried out for the past 10 years. Okay, that the first concept was, and the microbial desalination concept, okay, was introduced in 2009. Okay, after that lot of studies have been carried out. Okay, nearly 500 to 1,000 papers is available in this technology. Okay, this concept, microbial desalination concept. Okay, so many people have filed the patent also. So here this is one type of patent. Patent will be the tubular MDC and a quadruple MDC and a multi-stage MDC, upflow MDC and stacked MDC. Okay, these all are patented one. Okay, so similarly if you see it nationally, okay, nationally Dr. Gangrek has given he is a director of NIT Puducherry. So he is working in this field. Okay, so and similarly Dr. Venkata Mohan from the IICT Hyderabad and a few more Dr. Anil Verma from the IIT Delhi. Okay, they are working in this field, okay, microbial fuel cell field. Okay, they have filed their many patent. Okay, similarly we also filed the one patent. Like this is one type of patent which we have filed. So that patent we have the patent is also granted. Okay, so we have filed in the Indian patent. So we got the grant number, so 50504745. Okay, this is similar to the we have done. Okay, this has many advantages. Here you can see like it is highly compact, and here it can be minimal electrode spacing, and also it is we can have that is we can have options to place 2 or 4 anion exchange membrane or cation exchange membrane between the anode and the cathode, and it can be cost effective and it can be easy to scale up. Okay. So here we have that our system. So this is a conventional system. So here conventional system, as I told you, like anode and cathode and a middle chamber, that is a desalination chamber. So here this one, and this one will be that our patented one. Okay. So here multi-chamber MDC. Okay, our patented one we have done, and here we have done this different experiment. So here we have taken these 2 kind of the wastewater in these systems. So one is a black gum wastewater and sago effluent. Okay, these 2 type of wastewater we have used in the microbial desalination chamber. So this will be the conventional system. This will be the it is what we have patented. Okay, so here we have tested 2 different kind of the wastewater. One is a black gum wastewater and a sago effluent. Okay, so here we are given the conditions of the sago effluent and the black gum wastewater. So our main aim is to check that COD removal as well as the salt content in the saline water. So this is a COD efficiency, COD removal. So here COD value, and here so we have tested this conventional as well as the like a patented one. So we observed that maximum performance, okay, with respect to the maximum performance we observed with our patented one as compared to the controlled one. The control means conventional one. So here we observed the maximum power density of, it will be 0.25 milliwatt per meter squared, but as compared to this conventional one we got it very less number. Here you can see it will be around how much? It is a 0.20. So efficiency wise, if we see it, it will be, it's a 45% efficiency, that is COD removal efficiency 45% here, but here it will be we achieved this nearly 65 wastewater treatment efficiency, 65% we achieved the wastewater treatment. Okay. So similarly it is here this what is that? So conventional as well as this some kind of the like two different wastewater we are tested. Okay. So how far it is improving that based on the what type of wastewater used in this. So from this what we have concluded, like if I take the 2 different wastewater, so sago and the black gum wastewater. So here you can see we achieved this COD removal efficiency will be the 62%, but here in the sago effluent, so COD removal efficiency it will be 55%. Okay. So but to test this, so we have not used existing microorganisms. So because we have here so we are testing only the design based. Okay. So if I want to test the design based, so other parameters or other factors must be constant. Okay. That here what we did, we have kept constant of microorganisms. So we have isolated the one microorganism, or we have bought one microorganism from the one center. Okay, that microorganism used as a catalyst in this study. Okay, instead of going for the, because we are taking the 2 different effluent, no, like sago effluent as well as the black gum wastewater. So black gum wastewater may be the one type of microbes, contain maybe one type of microbes, but sago effluent may have the another type of microorganisms. Okay, so if we test in the both effluent in the one system, we cannot get the, if I get the different performance, we cannot justify this is because of that design aspect. Okay, because it's both will have the different microorganisms. To avoid that, whether it is purely our system is giving the better performance or not as compared to the conventional system. So what we did, so we have killed all the microorganisms from the both wastewater, and we have added the new microorganisms in the systems. Okay, that is Schwannella species we have added in the system. We have tested the performance. So hence we got the different kind of the performance here. Both we can see find out. Okay, we can see here. So this is one. Here this work we have published in the Journal of Water and Environment Research. Okay, so we observed this as compared to the SAGO effluent, okay, as compared to the black wastewater, we achieved the better performance in the SAGO effluent. Okay. So, this is one study. The another study is plant derived mediator. So, as I told you, okay, some bacteria can act as a mediator between the electrolyte and the electrode surface. Okay. Some bacteria may not be actively involved in that process. Okay. As I told you in that last yesterday's sessions, so what are the bacteria involved in this process? That bacteria should have the capability of transferring the electron from the electrolyte to the electrode surface. Okay. If that bacteria is not having the capability, then that bacteria will utilize the organic substrate for their growth only. It will not generate any electricity. Okay, any electricity. So, some kind of the bacteria itself it can transfer. Some kind of the bacteria we need to add some kind of the mediators. Okay, so the commonly used mediators are potassium ferric cyanide and thionine. Okay, so these two mediators, okay, these two potassium ferric cyanide and thionine, it's harmful to the environment. Okay. So, what we did, so we have taken this some mediator like natural mediator. Okay, natural mediator we derived from the mediator we derived from the naturally. Okay, different kind of the sources. So here we have taken the two different sources. One is vetiver extract and another one is the anion peel. Okay, these two extract. So these two extract we have used to extract the mediator from these systems. Okay. So these two mediator extracted from this and we are tested here. We compared the performance with that. Okay. So which one is giving the better, okay, as compared to the conventional also. So we don't want to go for the some conventional mediators. Okay. Conventional mediator because it is a chemical mediator is harmful to the environment. Okay. To avoid this we went this one like plant extract. Okay, plant based extract we have done this. So here you can see how far this it is influencing the performance with respect to the dosage of the, that is concentration of the extract or concentration of the mediator. Okay, so we have tested a very low quantity to the high quantity. Okay, it means that it's a 10 mg, 10 microgram, not mg, 10 microgram to the, it is 750 microgram we have tested. Okay, 10, 100 and 250, 500 and 750. Okay, like this it's tested. Okay, tested with both mediator. Okay, like vetiver extract and anion peel extract also we tested in this. So how far it is influencing the performance. So this paper we have this work. Okay, this work we have published in the Journal of Chemical Engineering. Okay, one of the most high impact factor journal. Okay, recently we published this work in that. Okay, so lot of work we are carrying out with like this similar here we have taken the only two kind of the extract, no, like vetiver extract and the anion peel extract. Okay, so now we are modifying this instead of another kind of the extract. Okay, instead of going for the waste, so waste based plant we are taking that. Okay, so we are doing a lot of work in this. So here you can see there will be a both extract. So what will be the performance? We improve with the literature reported. Okay, so here you can see it is the power density, how far it is improving with respect to the others reported in this. Okay, so most of that they are also trying this, but we achieved the better than the others work. Okay, 38.27 milliwatt per meter square power density we achieved as compared to the any other literatures. Okay, so we achieved the maximum COD removal efficiency of 92 to 97%, maximum COD removal, and similarly so desalination efficiency around 32%. Okay, as compared to the literature we reported. So then next we will go for the another types of microbial desalination cell. So like as you know this in the microbial fuel cell, so anode side it's a wastewater, but cathode side will be the aerolite. Okay, either the any water or pure water, that's deionized water, or we can take it buffer. Okay, buffer or it is aerolite. Okay, aerated cathodite we'll use it. Okay, so in the cathode side we need oxygen. Okay, we need oxygen. So if I how I can use the oxygen? Either the source will be either I have to use the pump, okay, to supply the oxygen to the system. Okay, so instead of what we can do, so we have used the algal based one. So algae, what will happen? What will do? So algae will consume the CO2 from the atmosphere and it will release the oxygen. Okay, that oxygen can be used for the in the cathode side to complete the cycle. Okay, so here so algae will have some important properties. Okay, as I told you, like it can be, so it can release the it can consume the CO2 and also it can release the oxygen, and at the same time that algae can be used as a biomass, biomass or substrate. Okay, because algae contains some kind of the carbohydrate, lipids and the proteins. Okay, lipids and the proteins will be there, that can be also used as a substrate in the microbial desalination cell. Okay, so either that algae can be used in the anode side, okay, anode side can be used as a substrate. Okay, if you use it in the algae in the cathode side, so it is used for releasing the oxygen. Okay, it is used for to supply the oxygen to the microbial desalination cell. Okay, microbial desalination cell. So here you can see, and also, so suppose if the same algae, so here what will happen in this? So it needs carbon dioxide. So algal growth, for growing the algae needs carbon dioxide. So how we will get the carbon dioxide? We cannot supply the carbon dioxide from the externally. Okay, it is again it is cost waste process. So, but at the same time, what will happen here? In the anode side, we are using the wastewater. That wastewater, it releases the, because of the degradation of microorganism degradation of organic substance by the microorganism, it releases the carbon dioxide, isn't it? That carbon dioxide passes to the cathode side. Okay, cathode side, that carbon dioxide will be utilized by the algae. That algae will release the oxygen for the reactions to complete the reactions. Okay, so and one more advantage is of by using the algae in the system. So at the end of the process, if you see it, it is algal will be there. Okay, that biomass can be used for producing the any kind of the value added product. Okay, that biomass, algal biomass, used for biofuel productions, or it can be used as a fertilizer, biofertilizer for the plant growth. Okay, so it can be used here. So this is one, and these studies based on the algae. So we have published our results in the one journal. Okay, Science of Total Environment. We have done different experiment with this, and we got these different results. How for this? Okay, so how for the wastewater concentration and the wastewater concentration, saline concentration, and the algal conditions, how it is influencing the performance of the microbial desalination cell. Okay, this one reported. This is another one. So bioelectrochemical system. So as of now, we have seen this bioelectrochemical system is mainly used for only the remove the salt. Okay, remove the salt content from the saline water. Okay, and wastewater treatment and the energy production. Only these three concepts only we have seen. Okay, microbial desalination cell or microbial field cell, or we can say bioelectrochemical system, can also used for removing the some metal ion, okay, from the wastewater. So if I take the battery wastewater, okay, or electrochemical industrial wastewater, it contains some kind of the metal ion like chromium or lithium or any other metals may be there. Okay, that metals can be degraded by this kind of the process. Okay, here you can see one of the example like chromium 6. Okay, chromium 6 is harmful to the environment, but at the same time chromium 3 will not be harmful. Okay, will not be harmful. So what the system can do? So the system can reduce the chromium 6 to the chromium 3 level. Okay, chromium 3 level. The same concept. So, but whatever the microorganism you have selected in this process, that microorganism should utilize the chromium. Okay, utilize the chromium or ions or any other content. okay, any other metals, okay, like ferrous also can be reduced. Okay. Based on that, we need to select the specific types of species in the anode side. Okay. It is not only, so if we take the specific species in the anode side, so that species will consume the metal ion, from the systems. So if the same kind of the metal ion containing wastewater, if you take it in the cathode side, because of the electron transfer, it will reduce into the another state. Okay. So chromium 6, if you take it in the anode side, so that microorganism may consume that wastewater, so which contain the chromium or some other content. Okay, it can reduce into the another level. Okay, another state. Suppose same wastewater, if I take it in the cathode side, because of the electron transfer between the anode to the cathode, so that valency will be reduced. Okay, 6 to the 3 level. Similarly, so we can take any kind of the wastewater in the either the anode side or cathode side. Here you can see cobalt may be harmful this level, so it reduces into the CO2 plus level, it may not be harmful. Similarly, chromium 6 is harmful, chromium 3 will not be harmful, and mercury 2 will be harmful, and mercury 0 level will not be harmful. Similarly, Ag plus and Ag in the ground state will not be harmful. Similarly, selenium 4 will be harmful, but selenium 0 level will not be harmful. Okay, it can be reduced into this one. Okay, selenium 4 to the selenium 0 level. Okay, it can be converted. So these are systems. Okay, many studies have been carried out in this. So we have done only one system, like we are tested in the chromium removal. Okay, chromium removal by using the microbial fuel cell. Okay, that work we have published in the one conference in 2015. Okay, it was published in the one special issue. Okay, one book chapter we have submitted. As of now, we have seen many types of bioelectrochemical systems. So here you can see it's based on the applications. Okay, based on the what type of applications involved in this process, we can name it as. Okay, one is microbial fuel cell. Suppose if I want to produce the electricity, that system called microbial fuel cell. So if we are getting the hydrogen, okay, that system called microbial electrolysis cell. So if produce the hydrogen, or if produce the hydrogen and the wastewater treatment and desalination, that is microbial electrolysis cell. Okay, so if I produce the electricity and the removal of salt water and the wastewater treatment, that system we can name it as microbial desalination cell. Similarly, microbial solar cell. Okay, if you produce the electricity and if you produce the wastewater treatment, and at the same time it needs some external power supply. So if you give the power supply through the some solar panel or something, that solar panel, then that system we can name it as microbial solar cell. Okay, like this there will be a n number of naming is available. Okay, there will be 48 types of bioelectrochemical system is available based on the applications. Okay, so if you are reducing the CO2, okay, so the cell that system name it as CO2 sequestration cell. Okay, because of it removes the CO2 from the wastewater. So CO2 sequestration. Similarly, other kind, if you use the algae, so algal microbial fuel cell. If you use the algae and producing hydrogen, algal microbial electrolysis cell. Okay, like this there will be 48 types is reported by the many researchers. Okay, so you can refer many books. If you have any questions, you can ask me. Yes.
Thank you, sir, for your presentation. I have one doubt, like how do we, you mentioned that we should identify bacteria which have the capacity to transfer the electrons. Yes. Like how do we identify, is there any assay or is there any specific group of bacteria which can do that, sir?
So as of now, people have reported the Schwannella species, okay, and E. coli species, and Pseudomonas species they have reported, but particularly if you want to test that species, you have to go for the cyclic voltammeter analysis.
Which one, sir?
Cyclic voltammeter analysis,
Okay.
electrochemical analysis, we need to do that. Okay.
Okay.
So during that electrochemical analysis, so we have to test with some control that is pure without any microorganism. With the microorganism, if you test it, okay, so you may get this some kind of the peak in between that. Okay. So we have reported how far it is influencing the particular species involved in the electrochemical activity. So without microbes, if you run this system, okay, so it will not give any kind of the peak in between the cyclic voltammeter analysis. Okay. If a species involved in the systems, so definitely it will be some peak in between this range. Okay, you can identify with that. Okay, so based on that we can confirm that, okay, the species, it has some kind of the activity.
And also it should have the resistance towards the sodium chloride, which is
Yes.
Thank you, sir, for a very nice talk. Sir, one question, one follow-up question from the ma'am's question, that why can't we have electrogenic species from a gene bank like MTCC or ATCC and can directly put
Yeah, in the last studies we have got it from the MTCC only. MTCC only. But every time we cannot maintain conditions of that particular species, no. So what we can do this, so we have the wastewater, okay, that wastewater contains some kind of the species, okay, group of species will be there, okay, among that group which one able to produce the electricity, that we want to find out. Once you find out, for example, if 5 species in the wastewater, okay, the species 1, 2, 3, 4, 5 species, so 4th species having the electrochemical activity. So what you have to do this now, so we have to find out how we have to enhance the, we have to increase the 4th species of, so how we have to enhance the 4th species to be maximum in the wastewater. Accordingly, we need to change the conditions.
So then that species has to be isolated and
we need to test, like, yes.
Need to be, yes.
So once if we isolate it, we know the conditions of the particular species. Okay, for example, nutrient conditions. How much nitrogen will be there, how much phosphorus will be there. We need to identify.
Complete medium.
Okay? Once you identify 20% or 40% something that, if you identify, then if you put it that conditions in the wastewater, if you maintain that condition in the wastewater, then automatically the particular species will grow more. This is also mixed culture, but particular species will grow more, and that it will act as a catalyst.
Sir, one more question. Sir, you mentioned that microbial fuel cell to microbial electrolysis cell to microbial desalination cells. Sir, is there anything like electrochemical refinery model can be there which can work like we can reduce the CO2 or we can produce some kind of
No, I am not getting your point. What are you asking? That different
like
types of
if
types is available.
If we are targeting like a biorefinery concept, so can such concept
No, we have not done with the biorefinery concept. Okay, we are working in only the lab scale model.
Yes.
So we are planning now.
So such kind of system that could give me a CO2 sequestration.
Yes, we are planning now. So we are working on that. Okay, instead of going for the single cell, we are going for the stack mode operation. Then after that, we need to do the some biorefinery concept based.
Yes, yes, so that we can have from energy to wastewater to desalination, all this in a series or linear based system.
We have to do that.
Thank you.
Yes, you can remove it. It's not only for salt removal, that is saline sodium chloride alone. Okay, the same thing we can use it for this magnesium or any other harness type of salt if we can use it. Okay, the same in the saline chamber we are taking the sodium chloride. Same in the saline chamber, if we take it potassium dichromate, okay, it will remove the chromium ion, will go to the one side and potassium will go to the another side. Okay, potassium will go to the cathode, chromium Cr minus ion will go to the anode side. Hmm. HCl will be in the anode, HCl will be in the anode side. Anode side. Sodium hydroxide will be in the cathode side. Yes, over the period of time it will affect. We need to replace it. We have to maintain the conditions of anode. So if HCl is producing, okay, so it is in the bacterial till the bacterial level is accepted, it will grow. Bacteria will grow. So if it beyond that limit, okay, for example, like E. coli bacteria will survive with the conditions of pH 7, okay, Pseudomonas and Shivonella species, okay, it will survive in the conditions of pH 6, okay. So if it goes beyond that level, pH 6 to the pH 4, then all bacteria will die. So we need to avoid that. So every time what we have to do, we have to maintain the pH conditions, or we have to done the experiment will be in the batch mode conditions. Okay, always of my knowledge, but I don't know exactly whether it is correct or not. All kind of the biological process will be in the semi-batch condition or batch type conditions. Even anaerobic digester also. Okay, it's a batch type only we can run. We cannot go for the continuous operation. 24/7 we cannot run it. Okay, into 365 days we cannot run it. Okay, at least 1 month we can run. Then after that we have to shut down that. Okay, or we have to remove the existing one from the reactor. Okay, in the anaerobic digester what they will do? So every 30 days they will load the substrate. Okay, 31st day they will load the first one. They will remove the first day one from the bottom. Okay, so that it will maintain these conditions. Okay, yes, the same concept here also. Any other? With this we'll stop. Yeah. Please ask. So any questions from the audience? Any questions from the online participants? Okay, no questions. Punit maina potukanga idhula pang chat box la pang. Second chat box. No audience. Okay. Okay, thank you.